I realize we're probably the last speakers between you and, if you were here for the robotics session, some heavy thought. So I will try to keep it brief. I'm glad you mentioned the two speakers. I was a bit afraid that I would be confused for Deanna. So I would be remiss if I didn't say this is a bit of a milestone for me. I spent, as of June 15th, I've been in the pharmaceutical industry for 34 years. And I left, most of that was at Roche, which is a good company. I enjoyed working for them. But they were big pharma, and therefore I'm a big pharma snob in most ways. And I have to say, my first PPMD conference was 5 years ago. And I really thought, geez, this little group of basically parent of DMD patients really think they're going to be effective. And after 5 years, if I have any expression that can be used sort of publicly, it's wow. I really think that PPMD in general and all the parent foundations really need to give themselves a hand or a pat on the back, because it's just, to me, miraculous and downright amazing what they have accomplished. I think, particularly dollar for dollar, orders of magnitude what Big Pharma has done for orphan disease or rare diseases. I'm here to talk today about the third leg of our development portfolio. It's AT300. The 300 is the third. I'll leave it as a discussion afterwards where we come up with the HT, DT, and AT prefix from. This compound, as are all of our compounds, I like to call them diamonds in the rough. They're compounds that I think normally wouldn't fit the filters that Big Pharma would use to select their leads. They have some, shall we say, warts or some unusual characteristics that a Big Pharma would stay away from. Some of the characteristics are in this slide that not necessarily themselves would be considered warts, but do describe what I think would be an interesting molecule, in my mind, a gee whiz molecule. It is a calcium channel modulator. It works on the stretch-activated channels. They tend to be overly active in DMD, particularly late-stage DMD muscle. They have been shown in a lot of preclinical tests. This molecule, which was originally discovered at the University of Buffalo, has been used extensively as a model for exploring stretch-activated calcium and its effect on, shall we say, normalizing calcium levels within a cell. And for those of you that are into electrophysiology, calcium influx and homeostasis are extremely important to normal muscle contractions. It's derived from tarantula venom, and if that doesn't gee whiz enough, I think most of you with a biologic background kind of realize that the Mother Nature is handed. That means that the chemistry that makes up proteins, the amino acids, the roughly 20 characters which make up the 8,000 proteins in the body, all have a handedness to them. They're L-amino acids. And after millennium of evolutionary development, the body's very good at managing proteins that are L-amino acids and can chew them up and recycle them and use the amino acids for other things. And the really interesting thing about AT300 is it's made up of R amino acids. And the L form of the compound and the R form of the compound, meaning using R amino acids, are equally potent. But because there are amino acids, the body has almost no idea what to do with this molecule. It can't really metabolize it in the normal ways. And this is really what makes it a drug. If it was an L amino acid compound, it would be taken into the body and eliminated or chewed up and recycled very quickly. As R amino acids, I won't really show the data today, but I can say that the R form in the MDX mouse, we can dose once every 4 to 5 days and maintain good levels, which extrapolates to humans once every 2 weeks, maybe once a month dosing, where if it was the L form, we'd probably have to dose once a month. So it's a gee-whiz molecule from many perspectives. I am going to just talk briefly about a few things. The original abbreviation for the molecule, it's been kind of a running joke about this at this conference about the names of molecules. The original name for this molecule is GS-MTX4. It doesn't really roll off the tongue, so we're going to call it AT300. And this slide is just the one I'll show to show its activity. If you do individual muscle fiber from the MDX mouse and stretch it a lot and therefore damage it, you can see you get a certain amount of percent of the control loss, probably about 70% loss. If you add other elements to block stretch activation, you can improve that, but with the addition of AT300, you can almost achieve exactly what happens if you don't have any calcium in the matrix, so you don't get the influx of calcium. So it tends to be a very potent inhibitor of the stretch-activated calcium channels. As I said, there's very good evidence of its in vitro efficacy. It's a moderate-sized amino acid, and there's been I wouldn't call it a GMP method, but a good chemical synthetic route. Preliminary safety studies of several species have shown no adverse effects from the compound. There was quite a bit of worry that since it is a calcium channel modulator that it may have unexpected effects, but in normal animals we don't see any of that. However, there has been, again at this conference, for those of you who watch talks, I think everybody's shown MDX data. It's kind of the industry standard. When the previous developers or the previous owners of the molecule ran their MDX study, it didn't work. And therefore, we said after we took it over, the first thing we had to do was determine, is it really a drug or not? Can we get it to work in an in vivo situation? And therefore, we designed this proof of concept study. I'm not going to dwell on it because it didn't work for us either. We decided to use exercise as a mechanism to make the MDX model more clinical or more human-like, to stress the animals, and it turns out that for who knows what reason, probably because the exercise damages the muscle, makes it leaky, we had really excessive exposure in the leg muscles, or excessive variance in exposure in the leg muscles from very low to very high, all but making a definitive study impossible. We did spend some time, I'm not even going to dwell on this, looking at other things, and we finally settled on a new strain of the MDX. I'm not sure how new it is, but it's really become popular in the past few months. It's called the MDX D2. Instead of being MDX mutation on the C57 black background that is normal, they used the DBA2 background, and the D2 is the Jackson Labs designation for that. And in fact, and then we don't do exercise, and we can show good exposure in all the different muscle groups. There we go. Oops, I did just what everybody else did. Yeah. So in a 2-week study, we were able to show good exposure across those types. I'm going to jump across this. At least I think I am. And then the other thing we did differently was, instead of looking at kind of standard performance measures which have a lot of variability like exercise tolerance, we went to an electrophysiology approach where you actually isolate the limb and you stimulate it and you look at loss of performance in a very controlled environment. In this particular case, you stimulate it once a second for 20 seconds, or 20 times, once a minute for 20 times, and then look at loss of performance. And there the black boxes are the control animals. The red circles are the untreated animals, and the blue triangles are the treated with the AT300. And you can see that there's, and this is after only 2 weeks of treatment. So one of the nice things about this compound, most other drugs for clinical trials for DMD need some time for remodeling to occur. So you need, one of the previous speakers had an 18-month clinical trial to see effect, but in fact this effect takes immediately. It's either going to work or not work in terms of calcium homeostasis. And so even after 2 weeks of treatment, we can see a very big impact on the muscle's ability to withstand stress. And then we did a 6-week pilot, and just quickly you can see again when you treat the animals, and here we've actually, for the first 2 weeks we do a loading dose where we dose every other day, but the other 4 weeks we only dose once every 4 days. Here we're looking at exactly the same experiment, but we're only looking at the first pulse and the last pulse, and you can see with the AT300 treatment that in fact there is a maintenance of the force in the red, and the middle slide is just a quantification of that maintenance change, the decline between the first and the last pulse. And you can see there's roughly a 70% loss without treatment, and only about a 50% loss. But most importantly, and I'm not going to show the slides for this, but we have some nice color graphs. We also give Evans blue dye for those of you that are used to giving dye to cells. If a cell is patent, if a cell is working well, it excludes the dye. But if a cell's sick, then the dye goes inside, and then you can quantitate how much of the blue dye is taken up as a measure of how healthy the cells are. And you can see on this right-hand graph is just a plot the percentage of total cells in the field of the microscope that are taking up the Evans blue dye, and you can see the untreated animals, it's about 22% of the cells take up the Evans blue dye, but in the treated animals it's only about 10%. So in summary, AT300 is a novel, unique, and well-documented target that plays a pivotal role in DMD pathology, that is loss of calcium homeostasis. The biology of the molecule has been heavily studied and published over many years, and assuming that now we've done all of our pilot studies in the D2 mouse, if we move on, or we actually just tomorrow will finish up our pivotal trial for the compound, and assuming that's the case, then we will start the normal IND studies, GLP studies, etc., for entry into clinical trials sometime in 2017. Thank you.
And I am the last speaker, so I'm going to show very little data. I'm going to show cartoons, if he puts my slides back. Can you put that back? Or we can go all to the pool. Yeah.
You don't want to go there.
And back, and back, and back. And that's the end. Cartoons. Alright, so you saw it all. Is it clear? Okay. Alright, so the third drug that we have is called DT200. And what it is a novel approach of making muscle bigger and stronger. So you have heard over 3 days, especially the first 2 days, about all the myostatin pathways and how that can turn muscle bigger. This is a different way of doing so. What DT200 is a selective androgen receptor modulator, or SARM. And basically what it is a drug that is similar to testosterone, right, the hormone testosterone. But does not act in every tissue that has androgen receptors. It is much more selective for muscle. And that is what we want, because what we want is this drug to act on the muscle, to increase muscle mass by increasing protein synthesis, and also in a testosterone deficient stage, which is small boys, this type of drugs also decrease muscle catabolism, so muscle degeneration. Okay, so through both mechanisms, it can make muscle bigger and stronger. And that, of course, that's really important in Duchenne, because as the muscle go through these cycles of necrosis and regeneration, the regenerated muscles are not normal. They are smaller, they are splitting, okay? So it's very important to help and act on those muscle fibers to make them bigger and stronger, but also to stop the deterioration of the muscle. These type of drugs also might have been shown actually to increase mitochondria in the muscle, mitochondrial biogenesis. And you heard the talks before about how important it is to have good mitochondria in the muscle, so that could be another benefit. So this is about what is androgens and the androgen receptors, and I think that most of you know what they are. They are hormones that play a critical role in all the tissues, and they regulate, of course, reproductive tissues and make boys boys and girls girls. But they also have effects on many other tissues, and we just go here. So some of the potential benefits of the drug is that it acts on the central nervous system and can increase cognition, energy, libido. It also acts on bone, increases bone formation, reduces bone resorption, and of course it acts on muscle and increases muscle strength. But if we let androgens, if we just will give somebody testosterone, it also could have some potential risk. The effects on those tissues will be okay, but in these ones it might not be okay. So acting on the bone marrow, it might increase the amount of blood cells, and when you do that, the blood becomes very viscous, very thick, and that can produce strokes and other issues. Of course, in males, it can act on the prostate, and that is a problem because it can make it larger and produce what is called prostatic hypertrophy. In the skin, it can, of course, make women have hair all over the place, and the same as boys, and cause balding. So what is important is to develop, and that's why this type of drugs have been developed to be selective for muscle, but try to decrease everything else. What makes this type of drug selective is not very well known. It could be because they distribute differently in different tissues, so you get maybe to muscle more than other tissues. Also could be because of the way they are metabolized inside the cell. Once of these drugs get into the cell, there are two types of machineries that can convert them, and that's going to make them more or less active. Okay. So are there other SARMs out there? The answer is yes. There are several, and there are several that are very close to the clinic, but the key of the question is the selectivity. A selective androgen receptor modulator is going to be a great drug if the selectivity is really high, because then the side effects are going to be very low. And what this drug has is one of the most selective of the SARMs that are there. And this is the only data I'm going to show you. What you see in blue and the open squares is the activity of this drug in both the prostate, so a sexual tissue, and a muscle. It's called the levator ani, okay? And as you can see, testosterone basically has almost the same effect both in the sexual tissue as it has in the muscle. But what you see in green is the prostate activity in the open circles and the muscle activity of DT200 on muscle. And as you can see, it has a tremendous amount of effect on muscle, not as much as testosterone, and almost none or very low in prostate. And that is what it makes it a really interesting drug. That's the selectivity, okay? And this is just in numbers. The VP over LA is this activity ratio between prostate sexual tissue versus muscle, and the higher that number, the more selective. On the right side, Osterine is the drug that is in phase 3 trials. And you can see the selectivity is much higher than the others, 220, but DT200 is about 1,000. Okay, so if this drug shows to have the same effect on muscle than the other drugs have, it has really a potential to be the best SARM out there. These drugs are being developed for people that have a small muscle, which are elderly individuals that get muscle atrophy, and people with cancer. That's called sarcopenia for the elderly people, cachexia for the people with cancer. And they have been shown to be effective, but of course the effectivity is limited, but the ability to increase the dose cannot be increased because they get side effects, because they are not selective. So we think that with our drug, because of the selectivity, we could have increased the dose enough to have a much more profound effect on the muscle. Our target, of course, is Duchenne muscular dystrophy, but if this drug works, of course, the big market is sarcopenia and cachexia. So what makes the main characteristics of this drug a great drug for neuromuscular disorders, not just for Duchenne, cachexia and sarcopenia, is the tissue selectivity, as we were talking. It's a once-a-day oral dosing, and it's not steroids, right? The athletes are taking this type of drugs, steroids, but they do have side effects. This is non-steroidal. Improves muscle mass, strength, and motor function in the MDX mouse model. And this has been shown, and of course in other type of muscle sarcopenia models, but this has been shown in the MDX mouse in the short term and long term, to increase the muscle mass and increase motor function. It has good safety profile, no hepatotoxicity, and minimal effects on hormonal status. The drug has been already in 3 phase 1 trials in normal human volunteers. And I wanted to show you that. So it has been on a single ascending dose in 28 males. It has been in multiple ascending dose phase 1 study in 24 males, and they checked 3 doses, 0.5, 1.5, and 5 milligrams per day for 2 weeks. And then in another multiple ascending dose in males and females, also over 7 and 14 days. And the dose that was chosen for that was the 0.5 milligrams per day. That is the dose that shows no side effects and no sexual adverse events. So that is the dose that we're going to move forward. The safety of the drug has been shown in these 78 subjects. There has been no serious adverse events, no discontinuations because of adverse events. Most of the adverse events were mild. They were unrelated to the drug according to the investigators, and this included headache and an increase in ALT. But not other liver functions, and that ALT appears to be from muscle. The third study did not look at DEXA or stir function or motor function like other drugs have done. What they did to look at the activity in humans was to measure with muscle biopsies something that's called protein fraction synthesis. And it's a way of knowing if there is actually production of new muscle, and that was positive. But because they didn't do DEXA, they didn't do stair climbing, which is what the regulatory pathway of these drugs is, we are going to conduct that proof of concept study. So we are going to be doing another phase 1 proof of concept study. There's going to be 4-week duration. It's going to be done in the UK, normal human volunteers, about 74 subjects. We're going to look at other 3 different doses versus placebo. And this is going to be launched probably late this year, probably before the end of the year. If that proof of concept study is positive, which means that we see increase in muscle mass index and increase in motor function, we're going to start on the DMD program next year. Okay.